In this lecture, we're going to be talking about how things move through the cell. So we have a whole bunch of cells in our heart, and they have membranes around them, and somehow things get into the cell and out of the cell. And that's what we're going to be talking about. So these PDF is there for you to share and use if you like. This is how this lecture fits into the overall discussion of cardiac electrophysiology. And as before, I'm going to ask you some questions to get you started. You might not know the answers to these questions at the beginning. You probably don't, otherwise you wouldn't be watching this. But when the lecture's over, you should be able to answer them very easily. So the first question is, where are sodium, Na, potassium, K, and calcium, which is Ca, normally found in the polarized cell? So when cells are in their resting state in the heart, there's sodium and potassium and calcium. Some of it's inside the cell normally, most of it, and most of the others are usually outside of the cell. So which are which? Where do you find sodium, potassium, and calcium in a polarized cell? Then I'm going to ask you a little bit about what a voltage-gated channel is, and how do voltage-gated channels become involved with depolarization. Similarly, what are G-coupled protein receptors, and how do they cause changes inside the cell? And then what is a ligand-gated channel, and how does it cause changes inside the cell? And give at least one example of a ligand. Okay. I'm going to talk about primary active transport pumps that move things inside and outside of the cell using adenosine triphosphate. They require energy in order to work. They're like powered pumps. And we also have ion exchangers. And we're going to talk about what the ion exchangers are and which ions they exchange. We're going to do a little bit of a deep dive when we talk about the ion exchangers, which we'll come back to later on to help reinforce it, talking about how acidosis inside the cell paralyzes one of those exchangers, and that can lead to irritable dysrhythmias in your patient. So you'll get a little impressive bit of knowledge when people say, well, why does hypoxia lead to ventricular irritability? We'll talk about the actual physiology of why that happens. And we'll talk about those exchangers, the two specific ion exchangers, and what are the ions that they're exchanging. Okay, movement through the membrane. So in a normal resting cardiac cell, the blue is the outline of the cell there, we have concentrations of potassium, calcium, and sodium. And in its resting state, you'll find that most of the potassium, I think it's like 98% or something, of our body's potassium is actually stored inside the cells. So we sequester potassium into the interior of our cells. And then floating around outside of the cells are two other ions, calcium and sodium. This is where they normally rest. Now, since there's so much potassium on the inside of the cell, you'll see there's a little purple arrow there saying electrochemical. What that's illustrating is that there's an electrochemical gradient that exists. With so much potassium inside the cell, the potassium would actually like to get out of the cell. So if we could find a doorway in the wall of the cell, in the cell membrane, and just suddenly open it up, the potassium would be going, oh great, we're crowded in here, and it would run through that open doorway just automatically. We wouldn't have to do anything. We wouldn't even have to exert any energy. We just have to open up the door, and the potassium will rush out of the cell along that electrochemical gradient. Similarly, with most of the calcium outside the cell and most of the sodium outside the cell, if we could somehow open up a door in the membrane, because those ions normally can't get through the membrane all on their own. They have a charge, the membrane is charged, so they repel, like two positive magnets. They push away from each other. But if we can open up a channel, the calcium will just rush through that channel and the sodium will rush through that channel into the inside of the cell. And if you keep the channel open, they'll do that until roughly there's the same amount of potassium inside the cell and outside the cell, and the same amount of calcium and sodium inside and outside of the cell. Of course, if we did that once and we couldn't reset the ions somehow, then we'd be in trouble because we wouldn't be able to fire our heart anymore. We wouldn't be able to fire our neurons anymore. And without our heart working and without our brain and our nervous system working, we're dead. So it's really important that we maintain these concentrations in the resting state, that we can exchange them to get something done, and then move them back to their original positions so that we can do it again. If we could only ever do this once, our heart would beat once and we'd die. We'd have one thought and then we wouldn't be able to do anymore. So having the ions in the resting state, switching, and then back again is an absolutely fundamental part of our normal physiology, and the ability to do that over and over and over again, to depolarize our heart 50 to 100 times a minute, has to continue throughout our entire life. In fact, by definition, when you stop depolarizing your heart and your heart stops, then you're dead. So this is the essential fundamental chemistry of life, moving ions around to activate our body systems. So how do they get in and out? If they can't get through the cell walls, what sort of channels do we have that allow the potassium and the calcium and the sodium to move around? Well, there's actually a bunch of different ways that things can get through the cell wall. And the rest of this lecture is going to talk about the sort of 4 major categories of ways that things get in and out of the cell. So if we take a look here, what we see is a diagram of our cell membrane, and there's 2 layers to our cell membrane. And on one side, we've got the outside of the cell, and on the other side, we've got the inside of the cell. And because there are a lot of things that can't travel through the cell membrane directly, so anything that has a very strong electrical charge, or anything that's too big, like glucose molecules or proteins, they're too big to be able to get through the cell membrane. So we've evolved to have these ion channels, and these channels are like portals or windows or doors through the 2 layers of the cell membrane, and when they open, they allow things selectively to go through the channel and move either from the inside to the outside or the outside to the inside. Normally these are closed, so things don't move automatically. We've got these closed channels or closed doorways, and when we signal them somehow, they open up, and that allows the ions to start to move. So let's go back to this diagram of the cell. Here we've got our cell, and we've got 3 channels, and they actually are of varying physical size. The potassium channels, the sodium channels, and the calcium channels. They're sitting there, they're closed normally, and they don't allow anything to go through them. These are called voltage-gated channels, and what that means is gating just means opening it up or closing it. And voltage gating means that when an electrical signal hits those channels, when we have an electrical charge going through the plasma membrane, it'll come along, it'll hit the potassium channel, for example, and the potassium channel will then open up and allow potassium to flow through. Now remember, where's most of the potassium? Question for you. Most of the potassium is inside the cell. So when we open up the potassium channel, when we voltage gate it, when we open it up with our electrical signal, and the channel opens up, the potassium that's inside the cell is going to run out. Another question for you, where's the sodium and the calcium? In its resting state, sodium and calcium predominate outside the cell. So when we open up, when we voltage gate those channels, the sodium is going to rush from the outside of the cell to the inside, and the calcium is going to rush from the outside of the cell to the inside. We also talk about these channels as being time dependent. So we'll trigger them with a bit of electricity and they'll open up, and they'll stay open either for a short period of time or a long period of time. The channels that open up for a short period of time are called fast channels, and the channels that open up for a long period of time are called slow channels. So they're sitting there, they're closed, an electrical signal comes along, zzz, they open up, sodium and potassium, calcium, whatever it is, rushes along the electrochemical membrane, whichever direction they go. They stay open for a little while, either fast or slow, and then they shut again. And it's very important that they shut, because when they shut, they need to reset before they can open again. So shutting our channel means there's going to necessarily be a bit of a pause, a refractory period, before we're able to open those channels up again. So that's the cycle we go through. Closed, zap, open, fast or slow, close, reset, and then zap, they're ready to open again. If they close and they get zapped right away, they won't open right away. There's this refractory period, this rest period that they mandatorily must have before they can open again. Okay. I've thrown this in just to give you an idea of the weird terminology that physiologists use sometimes. So we say that sodium is usually outside of the cell, and when we open up a sodium channel, obviously the sodium is going to rush into the cell along the electrochemical gradient. And you'll sometimes see that written in books or textbooks, chapters or articles or whatever, as I, N-A. And the I stands for, French, intensité du courant, which means the intensity of the current. It's French physiologists who discovered this, so they get to name it in their language. And the I is, the superscript I is just there for basically saying that's the intensity of the current. That's the direction of the flow of the current. So if you see the I and you're thinking, why would they call it I? Intensité du courant. That's where it comes from. Okay, let's leave our voltage-gated channels behind and move to a different type of channel. And those are called receptor-gated channels. Again, gated just means opening. So instead of opening due to an electrical signal, these channels have keys that come along, go into the lock, turn the lock, and that opens the channel up. And these keys, we talk about, the terminology we use in physiology is that they're ligands or ligands, depending on who you're talking to. So these ligands will come along, they'll enter into this lock and key mechanism, turn the lock, and boom, the receptors will open. So these are receptor-gated channels, not voltage-gated channels. And a lot of receptors exist in our body to open up a lot of different channels. One of the most common ones is insulin. So this is my channel. Here's the outside the cell, here's the inside the cell, and I've got these great big sugar molecules coming along, and they can't get through the plasma membrane. So my ligand in this case is insulin, and insulin comes along, hits the lock and key, turns the mechanism, that opens up the receptor-gated channel, and now my big sugar molecule can squeeze in and I can get sugar inside my cell so I don't die. There are hormones, there are steroids, there are prostaglandins, there's all sorts of different chemicals that act as ligands to open up these receptor-gated channels. But that's an important way, an important second way, that we get things from the outside to the inside of the cell. So, voltage-gated channels and receptor-gated channels. Under the receptor-gated channels, there's another type of channel as well, and these have a pretty fancy name. They're called G-coupled protein receptors. And these channels are a little bit different. With the receptor-gated channels, we get our ligand coming in, turns the lock and key, the channel opens, and something goes through. With G-coupled protein receptors, it's a little bit different. What happens is when we put the lock and key in there, when our ligand comes in and changes the morphology, the shape of the channel, a signal goes through, and that signal causes a whole bunch of chemical reactions to happen inside the cell with stuff that's already inside the cell. So let me draw a comparison. I think of voltage-gated channels as somebody with an electric car door opener. They just press the electricity and it opens. Receptor-gated channels are more like keys that will open up the whole car door itself. And the G-coupled protein receptors, I tend to think of as more like a doorbell. You come, you put the proper finger, the proper ligand to stimulate the doorbell, a signal goes through to the inside of the house, something secondary happens. In the case of a doorbell, the bell itself starts to go brrrr and ring, and then inside the house, inside the cell, things start to happen. People get up to answer the door, or we start to increase protein synthesis inside the cell, or we do something as a result of stimulating that doorbell. So there's all sorts of important ligands that work through this method, through the G-coupled protein receptors. That's why I'm mentioning it. It's not arcane. It's actually important. So neurotransmitters will work in this way. Adrenaline, which we use a lot, works in this way. Narcotics work on this way, on the mu receptors. That's the particular G-coupled protein receptors that narcotics work on. In fact, about a third of the drugs that we give to humans work on G-coupled protein receptors. So they're really important. When you're learning how fentanyl or morphine works, if you don't understand how G-coupled protein receptors work, then you're not going to be able to understand the basic physiology, because those are just examples of G-coupled protein receptor stimulation. Okay, so there's two types that we've done so far. We've done the voltage-gated channels and we've done the receptor-gated channels. Let's move on. And I'm going to talk again about the physiology. Let's just look at the sodium and potassium. That includes calcium, but for now, let's just focus on sodium and potassium as a simple example. When the electrical charge comes down to the voltage-gated channels, potassium channels open and potassium rushes out of the cell, sodium channels open and sodium rushes into the cell. It happens once and then we're stuck. Somehow we have to get the potassium back into the cell and the sodium back out of the cell in order to be able to do this over and over and over again, or else we die. And the way we do that is with what are called primary active transport pumps. And the big one that you've probably heard of is the sodium-potassium pump. So if we take a look at the diagram here, you'll see that we've got ATP coming down to ADP. Well, what does that mean? Our body has a battery system that it uses to run our body. So some of our cells charge these batteries, and then the batteries go out into the body and they're charged, and other parts of the body use those for powering processes like these pumps, and then the batteries have to get recharged. And here's how the batteries work. We've got this adenosine molecule inside of our body, and our body will take a bit of sugar and a bit of oxygen as a power source, and it will weld an inorganic phosphate onto this adenosine. So we come along, we breathe in some oxygen, we eat some carbohydrates or proteins or fats or whatever, and we use that energy to weld on one inorganic phosphate onto this adenosine, which we now call adenosine mono meaning one, adenosine monophosphate. If we weld another one on, now we've got adenosine diphosphate. If we weld another one on, we've now filled it up as powerful as it'll go, and this is our adenosine triphosphate. So adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. You can think of it as a battery with 33.3% charge, 66.6% charge, or 100% charge. That's the battery system of how our body powers itself. So this fully charged battery, adenosine triphosphate, comes along with its 3 phosphates welded on, and it comes up against one of these pumps. And when it comes up against one of these pumps, the pump goes, "Thank you very much, I'll take one of those," and it pulls off a phosphate. And in the process of pulling off a phosphate, it releases a whole bunch of energy. And so this pump here, we get our adenosine triphosphate coming up, we rip off a phosphate, adenosine diphosphate goes away, we've just reduced our battery power by a third, and then this pump starts to work. And you'll see that the way it's marked is that there's 3 sodiums that go out and 2 potassiums that go in. I like to think of these active transport pumps as revolving doors. They need a motor in order to work. And when we open our voltage-gated channels and potassium goes out, so we've got potassium out here, and sodium goes in, so we've got sodium in here, this is how we reset them. This revolving door turns once. And every time it turns once, 3 of the sodiums that went into the cell are kicked out, and 2 of the potassiums that went out of the cell are kicked back in. That takes energy. That's how we reset our cells. So the voltage-gated channels don't really take a lot of energy to work, but it does take energy to reset the system so that it's ready to go again. This sodium-potassium pump is incredibly important. We've got sodium-potassium pumps in our muscles, in our neurons, they're all throughout the body, so that when our brain fires off and neurons in our brain fires off, we reset them using the sodium-potassium pump. When our heart beats, we reset our cardiac cells using the sodium-potassium pump. In fact, about a third of all of the energy used from our batteries, our adenosine batteries are used to power the sodium-potassium pump. And we've got squillions of sodium-potassium pumps. Just in a myocardial cell, in a heart cell, there's 1,000 pumps per square micrometer, and a micrometer is incredibly tiny. I can't imagine this, but just we've got lots and lots and lots of sodium-potassium pumps. So that's our first active transport pump. What about calcium? I hear you asking. Well, there's an answer for that. The other active transport pump that we have is the calcium ATP pump. Now this one is another revolving door, but it doesn't actually kick anything into the cell. It just kicks calcium out. So you can see the system here. We open up the voltage-gated channels, potassium rushes out, sodium and calcium run in. Then we fire up the pumps using our ATP batteries. We kick the sodium out, we put the potassium back in, and we kick the calcium out. So the cell has now reset. So that's the main function of what I want to get across. But there's one more sort of fold into this whole story. We've got our voltage-gated channels, we've got our receptor-gated channels, we've got our pumps, and we've got one more class of ways that things get in and out of the cells, and those are called our ion exchangers. So when we use our ATP pumps, we kick a lot of sodium back out. In fact, we kick out so much that we get a really good concentration of sodium on the outside going again. And we can use that high concentration of sodium on the outside to actually do some exchanging across this membrane, just sort of automatically based on the concentration gradient of the sodium. So the first one we have is the sodium-calcium exchanger. The calcium pump that we have doesn't quite get enough calcium out of the cell. So we get a bit of an extra boost by using this sodium, putting 3 sodium in, and for every 3 sodium we put in, again, this is like a revolving door, but the way I like to think of it more is instead of a revolving door, think of it this way. Think of it like a judo flip, or somebody doing square dancing, where you take your partner, you grab onto them, and then you both just switch position. This doesn't actually take energy because you're just using the concentration that exists. So we don't need to use ATP for this. There's a ton of sodium out here, so the sodium just grabs the calcium, 3 sodiums, grab a calcium, and they just, without extra energy, exchange positions, and that ends up driving a whole bunch of calcium out of the cell. Now the sodium, which goes back into the cell, needs to be kicked out again, and the way it gets kicked out is by going back up here to the ATP pump and the sodium-potassium ATP pump, and we kick the sodium back out again. And then the high concentration here pushes the sodium in, which kicks the calcium out. This sodium comes back up to the sodium-potassium pump and goes round and round and round. And the power that we generate from the sodium-potassium pump is used up here to help kick some calcium out. So that's our first exchanger. Now, here's an important part, and this gets a little bit funky, and you might not be able to hold on to this one right now, but just hear me through and it'll make more sense later. This sodium-calcium exchanger is inhibited when the cell becomes acidotic. So when we become distressed, and we're not able to breathe properly, we're not able to get enough oxygen into our cells, and they can't work properly, then the hydrogen starts to build up inside the cell becomes acidotic. And when the cell becomes acidotic, this exchanger doesn't work very well. It's paralyzed, it's inhibited. So that means we start to get a whole bunch of calcium building up inside the cell. And that's a problem. Because when we talk about the ions moving, we're talking about the electricity of the cell, but we're not talking about the actual mechanical contraction. We'll talk about this a little bit later, but I'll give you the heads up to say it's the calcium that goes into the cell that causes a whole cascade of things to happen, which makes the cell physically contract, physically get smaller. So the calcium stimulates the physical contraction. And when we have too much calcium here inside the cell, it makes the cell contract spasmodically and erratically. So it's a bit like your heart cells start to have these little seizures because there's too much calcium in them, stimulating the muscles to physically contract. So when we, for example, and this is heinous, you'd never actually do this, but if you were to do a horrible, horrible experiment, you could tape up somebody's face, make them stop being able to breathe, and watch on the cardiac monitor, and they'd start throwing PVCs. And the reason that happens is because we're getting a buildup of calcium inside the cardiac cells because the cells are getting acidotic. And this pump, or rather this exchanger, doesn't work anymore, and we start getting irritability. When somebody is hypoxic and they're not getting enough oxygen, then that also affects the neurons in their brain. This exchanger and others start to run into problems, and our brains start to depolarize erratically, and that's how hypoxia can lead to seizures, because our brains just start firing off. Okay, that's the first exchanger. The next exchanger is the sodium proton or sodium hydrogen exchanger. When we just have one charged hydrogen, that's just a proton. So we take advantage of that sodium gradient, push some sodium in, exchange it with a hydrogen that goes out, and that's how we keep the inside of the cell from becoming acidotic, because hydrogen is acid, basically, in simple terms. So we push a sodium in, throw a hydrogen out, take two of these hydrogens that have been thrown out, bond them with oxygen, and we now change this dangerous hydrogen into water, which we can easily excrete from our body. This sodium goes back up to the sodium-potassium pump, gets exchanged using ATP, comes back down. The concentration here drives more hydrogen out, also drives more calcium out, and these exchangers can work because of the concentration of sodium out here, which we're developing with our pump. So here's the really important thing you need to understand about the physiology of our cells. We've got ions and large molecules on the outside and some ions on the inside of the cell, and we need to exchange them and we need to put them back in order for our metabolism to work properly. So we've got four basic ways to do that. We've got the voltage-gated channels, we've got the receptor-gated channels, we've got the active transport pumps that rely on ATP in order to work, and we've got the ion exchangers, which use the concentration of sodium in order to get things to work. This is a fundamental diagram, and it's really important throughout our body. It helps understand how neurons work. It helps understand how muscles contract throughout our body. So when we give drugs, for example, that bronchodilate people, it's working on these systems. So understanding this is absolutely fundamental to understanding how physiology in general works. You should be able to take a blank piece of paper and write this diagram out, and that will really solidify some basic knowledge that you need to know for tons and tons of applications in physiology. Okay, so make sure you know this stuff. Get comfortable with it. I'd recommend you study it a little bit before you go on to the next lecture, so you've got it consolidated. If you've got any questions, leave them in the comments, or if you're one of my students, email me, and happy to discuss it.